photosystems consist of arrays of chlorophyll molecules. Chlorophyll, the green pigment of leaves, absorbs light energy. The absorbed energy excites electrons to a higher energy level. Energized electrons from photosystem 1 are passed down an electron transport chain and added to NADP+ to form NADPH. Meanwhile, energized electrons from photosystem 2 are passed through another electron transport chain. Their energy is used to pump hydrogen ions from the stroma into the thylakoid compartment, creating a concentration gradient. Electrons leaving this electron transport chain enter photosystem 1, replenishing its lost electrons. Photosystem 2 replenishes its electrons by splitting water. Hydrogen ions and oxygen are released into the thylakoid compartment. This is where the oxygen gas generated by photosynthesis comes from. The buildup of hydrogen ions inside the thylakoid compartment stores potential energy. This energy is harvested by an enzyme called ATP synthase. As hydrogen ions diffuse through ATP synthase down their concentration gradient, the enzyme uses the energy of the moving ions to make ATP. Next, ATP and NADPH are used in the sugar-making process of the Calvin cycle.
Carbon fixation occurs during the light-independent reactions, also known as the Calvin cycle. These reactions take place in the stroma, the space outside of the thylakoids. A 5-carbon sugar, ribulose biphosphate, RUBP, binds to carbon dioxide, producing an unstable 6-carbon sugar. The enzyme catalyzing this reaction is rubisco. The unstable 6-carbon sugar breaks into 2 3-carbon molecules. For every one of these 3-carbon molecules that eventually leaves the Calvin cycle as sugar, 5 are used to regenerate RUBP. ATP and NADPH provide the energy for the necessary rearrangements of molecules in the Calvin cycle.
Sugar during photosynthesis. This sugar is stored in the leaves as starch. To test the leaf for starch, the first step is to kill the leaf. The leaf is killed by immersing it in boiling water for a few minutes. The flame is turned off and the leaf is removed. The leaf is then placed in a boiling tube of alcohol. The boiling tube is then placed into a beaker of hot water. The alcohol begins to boil and the leaf slowly loses its green color. The alcohol is poured away. The leaf is then placed back into the hot water. This is to soften it. The leaf is placed on a white tile. The iodine test for starch can now be carried out. The leaf is stained dark blue. This indicates the presence of starch.
In this practical, we will determine whether chlorophyll is indeed necessary for photosynthesis. For the practical, you would need a de-starched leaf which is variegated, which means that there are parts that are green, which contain chlorophyll, and other parts, if you look on the margin here, you will see that no green pigment occurs in that leaf, and that's why it is colorless. Now, what do we need to do to de-starch this pot plant? We would have to place it into a dark cupboard for about 48 hours to ensure that all the starch that has been produced previously will have been used up, even the parts that would maybe be in the non-colored parts of the leaf. Once we have left this pot plant outside for a few hours, I will then pick one of the leaves and I will test for the presence of starch as shown previously in the first practical demonstration. Now, once we have done that and we have tested for the presence of starch and we have added iodine solution to the leaf after the test for starch, we will see that the parts that did contain chlorophyll has now tested positive for starch. But on the margin, where we see that no chlorophyll was present, we will see that the iodine solution has stayed its brownish color. The middle of the leaf, if we compare it to this leaf, shows that the parts that contain chlorophyll have turned to a darker color. Now, I have placed the iodine solution onto this leaf, and the longer I leave.
Carbon dioxide is essential for photosynthesis. Here is a small activity that will demonstrate that carbon dioxide is essential for the process of photosynthesis. Select a potted, healthy plant with long and narrow leaves. Keep it in the dark for 2 or 3 days. This will ensure that the leaves are free from starch. Now, take a wide-mouthed glass bottle with a cork to close the mouth of the bottle. Split the cork vertically. On the day of the experiment, take 5 to 10 milliliters of potassium hydroxide solution in the bottle. Potassium hydroxide absorbs the carbon dioxide present in the bottle. Early in the morning, insert 1/2 of a selected leaf into the bottle through the split cork without cutting it from the plant. The other half of the leaf, or the base part, is to be left outside the cork. The bottle is to be kept airtight, which can be achieved by applying grease or Vaseline around the rim of the mouth of the bottle. Make sure that the outside air does not come in contact with the leaf in the bottle. Keep this unit in the sunlight for 3 to 4 hours. After 3 to 4 hours, detach the leaf from the plant and take it out of the bottle. Test it for starch with iodine solution. We'll observe that the part of the leaf which was inside the bottle does not turn deep blue. However, the part of the leaf that was exposed to the surroundings prepares starch and so turns deep blue. Note that both halves of the leaf receive the same amount of sunlight and water, and both have chloroplasts. However, the portion of the leaf that was inside the bottle could not receive carbon dioxide. This proves that green leaves can perform photosynthesis only in the presence of carbon dioxide.
How can you prove that sunlight is essential for photosynthesis? The aim of experiment to prove that light is necessary for photosynthesis by black paper experiment. So for this apparatus required a potted plant, black paper or any screen or S-shaped or any design cut and iodine solution. Procedure. Take a plant with a discharged leaves, cover one of its leaf with the black paper on which design is cut. Fix the paper on a leaf in such a manner that light does not enter the dark part. Place the plant in sunlight. After a few hours of exposure to bright sunlight, test the leaf which is covered by the black paper for the presence of starch. So what will observe after testing? It is observed that only the parts of a leaf turns bluish black which could get light through cut design, showing the presence of starch. The remaining leaf will not show the existence of starch. What will conclude? The results. You can say this experiment process, that is light is essential, or you can say necessary to form a starch in a plant.
Let us see about an experiment on photosynthesis. The test tube and funnel experiment. The test tube funnel experiment demonstrates that oxygen is evolved during photosynthesis. A few branches of hydrilla are kept in a beaker containing pond water in which a small amount of sodium bicarbonate is dissolved. The branches are covered with a glass funnel and a test tube full of water is kept inverted over the stem of the funnel as shown in the diagram. Now the apparatus is kept in sunlight for 4 to 6 hours. The gas bubbles may be observed from the ends of hydrilla branches kept within the glass funnel. These gas bubbles are collected in the test tube by the downward displacement of water. The gas is tested for oxygen. When a burnt splinter is taken near the mouth of the tube, it glows brightly and proves that the gas is oxygen. The test tube and funnel experiment demonstrates that oxygen is evolved during photosynthesis.